Welcome, everyone, to our journey into the fundamentals of biomolecules. Today, we are going to explore a truly fascinating topic, the bonds that stabilize protein structure. You might not realize it, but inside every cell of your body, millions of tiny protein machines are working tirelessly to keep you alive. But how do these microscopic machines know what shape to take? And what holds them together? By the end of this lecture, you will understand the invisible chemical forces that fold a simple chain of amino acids into a complex, life-sustaining structure. Let us dive in and discover the architectural secrets of life. To understand how a protein gets its shape, let us start with a simple analogy. Imagine you have a long piece of string, and attached to this string are small patches of Velcro, tiny magnets, and spots of glue. If you toss this string into a washing machine and let it tumble around, it is not going to stay as a straight, flexible line. Instead, the magnets will snap together, the Velcro will catch, and the glue will stick. The string will crumple into a very specific, tightly packed three-dimensional shape. This is exactly what happens to a protein molecule inside your cells. It starts as a long, flexible chain of building blocks called amino acids. But to do its job, it must fold into a precise shape, guided by chemical bonds acting just like those magnets and Velcro. Now that we have our analogy, let us look at the actual chemistry. In biochemistry, the bonds stabilizing a protein are categorized into covalent and non-covalent interactions. The foundation, or primary structure, is held together by strong covalent peptide bonds, linking amino acids in a specific sequence. However, the complex secondary, tertiary, and quaternary structures are primarily stabilized by a multitude of weaker non-covalent forces. These include hydrogen bonds, hydrophobic interactions, and van der Waals forces. Additionally, strong covalent disulfide bonds can form between specific cysteine residues to provide extra structural reinforcement. It is the cumulative power of thousands of these weak interactions that truly dictates the final folded form of the protein. Understanding these stabilizing bonds leads us to a crucial concept, the native state. Together, all these bonds ensure the protein reaches its lowest energy, most stable conformation, known as the native state. This is absolutely critical because a protein's structure directly determines its biological function. If these delicate bonds are disrupted by heat, acid, or chemicals, the protein unfolds in a process called denaturation and completely loses its ability to function. For example, the enzymes that digest your food rely on their specific shape. Furthermore, if a mutation changes just one amino acid, it alters the bonding pattern and causes the protein to misfold, which is the root cause of genetic diseases like sickle cell anemia and Alzheimer's disease. So, we know that proteins must fold to function, but what drives this process physically? The overall stability of a folded protein is governed by the strict laws of thermodynamics. When a protein folds, it forms many stabilizing bonds, which is a favorable process. However, it also loses a massive amount of flexibility, becoming highly ordered. This creates a constant thermodynamic battle between minimizing energy through bond formation and maximizing disorder, or entropy. To understand if a protein will actually stay folded in its native state, we cannot just guess. We must look at the mathematical relationship between these competing forces to see which one wins. This thermodynamic battle is perfectly captured by the Gibbs free energy equation. Let us break down what each symbol means. Delta G represents the change in Gibbs free energy, measured in joules per mole. For a protein to fold spontaneously and remain stable, this final value must be negative. Delta H stands for the change in enthalpy, which reflects the heat energy released when stabilizing bonds are formed. A negative delta H is favorable. T is the absolute temperature in Kelvin. Finally, delta S represents the change in entropy, measuring the disorder in the system. Because folding restricts movement, delta S is negative, which opposes folding. The energy gained by forming strong bonds must be large enough to overcome this loss of flexibility. Now that we have our formula, let us see this with a concrete example. A biochemist is studying an enzyme at room temperature, 298 Kelvin. The formation of bonds gives an enthalpy change of negative 250 kilojoules per mole. The entropy change is negative 0.6 kilojoules per mole Kelvin. We need to calculate the Gibbs free energy change to see if the folded protein is stable. We substitute our values into the equation. We carefully multiply the temperature by the negative entropy change, giving us negative 178.8. This represents the energy penalty for becoming highly ordered. Subtracting this from the enthalpy, we get a final delta G of negative 71.2 kilojoules per mole. Because this value is negative, the folded enzyme is highly stable. We have just seen how overall stability is calculated, but let us zoom in on one of the most important specific interactions contributing to that stability, the ionic bond, also known as a salt bridge. This occurs between oppositely charged amino acids. Imagine two powerful magnets pulling toward each other. This is exactly how a positively charged amino acid interacts with a negatively charged one. However, the strength of this attraction is not constant. It is highly dependent on the environment surrounding the bond. A salt bridge on the wet surface of a protein behaves very differently than one buried deep inside the dry core. To understand this, we need to describe this attractive force mathematically. Before we look at the math for salt bridges, we must understand a crucial environmental factor: the dielectric constant. The environment surrounding an ionic bond drastically affects its strength because different mediums shield electrical charges differently. This shielding effect is measured by the dielectric constant. For instance, water has a very high dielectric constant. If a salt bridge is on the surface of the protein, the surrounding water molecules will shield the charges, severely weakening the bond. However, the inside of a folded protein is a dry, hydrophobic core with a very low dielectric constant. Because there is no water to interfere, salt bridges buried deep inside the core are significantly stronger and provide immense stability to the protein. Now that we understand the role of the environment, let us look at the mathematical formula that brings all these variables together: Coulomb's law. This equation calculates the electrostatic energy of the interaction, denoted by E. The constant K ensures our units align correctly. The variables Q1 and Q2 represent the electrical charges of the interacting amino acids. The symbol D is the dielectric constant we just discussed, sitting in the denominator. Finally, R is the distance between the two charges. Notice the mathematical relationships here. If the distance R decreases, or if the dielectric constant D is very low, like in the protein core, the denominator shrinks and the attractive energy E becomes much stronger, pulling the protein tightly together. Let us apply Coulomb's law to a specific salt bridge. Imagine a positively charged lysine with a charge of +1 and a negatively charged glutamate with a charge of -1. They are located deep inside the hydrophobic core, separated by a distance of 3 angstroms, with a dielectric constant of 4. We want to find the proportional electrostatic energy, ignoring the constant K to focus on the variables. We plug in our charges, the dielectric constant, and the distance. Multiplying the denominator gives us 12, and the numerator is -1. The final calculated proportional value is -0.083. This negative energy confirms that energy is released, providing a strong attractive force that locks the protein structure in place. Now that we have calculated the forces, let us look at a visual summary of everything we have discussed. On screen is a schematic of a folded protein chain. Notice the 4 distinct types of interactions holding the loops and helices together. On the top left, you see a strong covalent disulfide bond, forming a permanent bridge between 2 sulfur atoms. On the bottom left, delicate hydrogen bonds connect different parts of the protein backbone. On the top right is the ionic bond, or salt bridge, we just calculated, showing the powerful attraction between positive and negative charges. Finally, on the bottom right, hydrophobic interactions force nonpolar groups to cluster together, hiding away from the surrounding water molecules. All these work in perfect harmony. We have covered a lot of ground today, so let us review the key takeaways. First and foremost, a protein's ability to function depends entirely on its specific 3-dimensional shape. While strong covalent peptide and disulfide bonds provide the primary framework, it is the cumulative power of thousands of weak non-covalent interactions, like hydrogen bonds and salt bridges, that truly dictates the final folded form. We also learned that folding is a delicate thermodynamic balance, where the energy gained from bonds must overcome the loss of flexibility to minimize the overall free energy. By understanding these stabilizing forces, we gain profound insight into how life functions at the molecular level, empowering us to design life-saving drugs and intervene when proteins misfold in disease. Thank you for joining me in this lecture.